2 days before ovulation, the Graafian follicle was mature enough and ready to ovulate. It contains a primary oocyte surrounded by a group of granulosa cells known as cumulus. This collection is known as cumulus oocyte complex. Now let's examine the cumulus oocyte complex. The primary oocyte is present in the center, surrounded by an outer membrane known as cytoplasmic membrane. Also, it's called vitelline membrane. It contains a cytoplasm and a nucleus known as germinal vesicle. The oocyte is surrounded by an outer shell known as zona pellucida. As you see, there is a narrow space between the zona pellucida and the primary oocyte, known as perivitelline space. Around the zona pellucida, there is a collection of granulosa cells known as cumulus. The innermost layer of the cumulus is known as corona radiata. Corona radiata communicate with the primary oocyte through a fine connections called gap junctions. Inside the cytoplasm of the oocyte, there are many granules located on the cortical part of the cytoplasm and called cortical granules. They play a very important role in preventing polyspermy after fertilization. The germinal vesicle is eccentric in location. I mean, it is located peripherally and not in the center of the oocyte. This location of the germinal vesicle will establish the polarity of the oocyte. Now the oocyte has two poles. One pole towards the germinal vesicle, known as animal pole, the other one on the opposite side, known as vegetal pole. This polarity ensures unequal division of the oocyte. About 36 hours before ovulation, LH will increase sharply. This will result in resumption of meiosis. The primary oocyte was arrested in the first meiosis, and that's why its name is M1. Now it will complete the first meiosis to form a secondary oocyte and a first polar body. The secondary oocyte contains almost all the cytoplasm and its organelle, while the first polar body contains nearly no cytoplasm. LH will also trigger ovulation, where the oocyte and the part of the surrounding cumulus will be pushed outside the ovary and into the fallopian tube. As you see, there are three types of oocytes. Germinal vesicle, where the nucleus is visualized inside the cytoplasm. M1 oocyte, where no germinal vesicle visualized and no polar bodies. M2 oocyte, where no germinal vesicle is visualized inside the cytoplasm and a first polar body is seen inside the perivitelline space.